G'day, I'm Alicia Stewart and I'll be talking about ecosystem condition monitoring in temporary rivers in Queensland, Australia, using ants, plants and fish. And I'd like to thank my co-authors Jonathan Marshall and Peter Negus. Temporary rivers, rivers that cease to flow and dry up, are common worldwide. When they are dry, traditional indicators of river health, such as fish, water quality or aquatic invertebrates, can't be sampled. The sections of the stream that are sampled in a temporary river network during dry times and the resulting ecosystem health results do not represent the entire catchment. Well, so what? Who cares about dried up rivers? Well, they have human and ecological values that have previously been overlooked. They have a unique terrestrial invertebrate diversity. They can be egg banks and a refuge for specialised aquatic biota. They can be dispersal and migration corridors for terrestrial biota, sites for the storage and processing of organic matter and nutrients, and they have human values. In Australia, approximately 70% of the 3.5 million kilometres of river channels are intermittent, and the proportion of intermittent rivers in the global river network is likely to be over 50%. Many rivers that were previously perennial are now intermittent because of water management, so water abstraction, interbasin transfers, and also climate change. So monitoring only the wetted parts is not representative of the entire catchment. I work for the Queensland State Government in a group that monitors and assesses the health of riverine ecosystems. We incorporate all phases of temporary rivers, be they wet or dry, into our monitoring and assessment framework. Now, before I talk more about how we incorporate temporary rivers into monitoring and assessment, I'll do a brief run through of our framework. My colleagues listed here have written a book chapter on this, so please refer to the chapter for more detailed information. Firstly, we start with a list of generic stresses that are relevant to the riverine ecosystems of Queensland, and each is considered a potential threat to river health. We then choose our focus region that we're interested in monitoring. So this is a list of generic threats to riverine ecosystems that we've identified for Queensland. We've developed cause-effect conceptual models for each of these. Next, we engage experts to provide information on each stressor in the region. An ecological risk assessment for each threat was undertaken using information gleaned from the literature and expert opinion relevant to the focus region and threat. Risk is determined by the likelihood of the stressor occurring and the consequence of the threat if it does occur. We review the results to produce an initial desktop assessment of risk to river ecosystem health posed by each stressor. The stressors are ranked by risk, and that determines which ones will be assessed by targeted monitoring. This is an example of the outcomes of a risk assessment that we've previously done. You can see that in this example, introduced aquatic fauna and deposited sediment in the red column were ranked as high-risk threats to riverine ecosystems. Introduced riparian fauna in orange was ranked as a medium risk, and the other threats were ranked as a low risk. We then consider the resources we have and refine our conceptual models relevant to the focus region. This is a cause-effect conceptual model of threats from introduced aquatic fauna that we've made relevant to our focus region, the Queensland section of the Murray-Darling Basin, by adding introduced species that are found there and their reported and expected impacts. Indicators for each of the stresses and ecological responses to the stresses to be monitored are chosen based on those conceptual models, and sites are selected. The data we collect from sampling is used to assess the intensity of measured threats and ecosystem condition in relation to measured threats. This is reported along with management recommendations to mitigate the threats where necessary. So now I'll talk about an example of our work that uses this monitoring and assessment framework for temporary rivers. Our focus area is the Queensland portion of the Murray-Darling Basin, consisting of 4 catchments, the Bulla, Paroo, Warrego and Kneebine. As the title of my talk suggested, the indicators we chose were ants, plants and fish. We chose ants and other terrestrial invertebrates to measure ecosystem responses to the threat of introduced riparian fauna, such as feral pigs and cattle. Terrestrial invertebrates can be sampled at dry sites and also sites with isolated waterholes, as seen in the bottom right photograph, where most of the riverbeds dry. We use pitfall traps to collect the invertebrates, we identified them in the laboratory, and we calculated taxon richness and abundance. We also collected data from transects along the bank and in the riparian zone to measure the intensity of damage caused by introduced riparian fauna. From these data, we calculated pugging, rooting, and wallowing damage metrics. We chose plants, or rather introduced riparian flora, or weeds, as an indicator of introduced riparian flora impact. The spread of pest riparian flora has been linked to introduced riparian fauna, such as pigs and cattle. They can be sampled at all sites, whether wet or dry. We used 4 200-metre transects, 2 from each bank, taken perpendicular to each waterhole, and we took measurements from 20 quadrats per transect. Measurements were taken to record whether exotic floral species were present and what percentage of the flora consisted of exotic species. We chose fish, in particular introduced fish and other introduced aquatic fauna, to measure the intensity of the threat that these species pose and as an ecosystem response metric. They can of course be sampled at wet sites only. We used a combination of sampling methods, fyke nets, seine nets and opera house traps. We calculated the presence of exotic faunal species and the proportion of the total fish biomass consisting of exotic species. These are a couple of examples of just some of the results that were used in the assessment ratings for reporting. We've got terrestrial invertebrate taxa richness, the biomass of introduced aquatic fauna versus native fish, and the presence of introduced riparian flora. Looking at terrestrial invertebrates, you can see that the taxa richness range from 7 up to 19 throughout the catchments, and there's a bit of variation there with the highest richness recorded in the Kneebein catchment. Looking at biomass of introduced aquatic fauna, you can see that there's one catchment, the Buller, that has no exotic fish recorded. Exotics recorded in the other catchments include goldfish, gambusia and carp. Now looking at the presence of introduced riparian flora, again there's a catchment with no exotics recorded, and that's the Paru. There are weeds recorded in the other catchments such as mesquite, Parkinsonia, Negura burr, Mexican poppy, introduced acacia species and also prickly pear. I'll talk now about assessment ratings for the threat and condition categories. Note that these assessment ratings are arbitrary and are used as a communication tool only. Current threat was based on field collected data that described the current level of the threat to riverine ecosystem condition in the region from low to high. The current threat score is a validation of the risk score and calculated by direct field measurements. It may therefore differ from risk. The condition of riverine ecosystems was assessed using the field collected data that measured the ecosystem response to each threat. Each threat was assessed by comparing the values from all sites against a range of values represented by the reference sites, or compared against the value of 0 for those indicators that do not require a reference range. Values within the reference range were given a disturbance score of 1, being good condition, whereas values outside the range were allocated a standardised score between 0 and 1, equivalent to high, moderate or slight disturbance. These are the results as they appear in our government report on the right for the stresses of introduced aquatic fauna and introduced riparian fauna for each of the 4 catchments. You'll remember that the risk score for each stressor was determined from expert opinion that formed our initial risk assessment. The current threat is a validation of the risk score informed from field data collection. The condition of riverine ecosystems was assessed using the field collected data that measured the ecosystem response to each threat. I've listed a couple of journal articles there if you'd like more information, and also the book chapter on the risk assessment framework. Thanks for listening.